We're just going to do a quick overview of transcription and translation. So if you remember, the central dogma of biology talks about how we go from the DNA to making the protein. And remember, it's a two-step process. So first we're going to transcribe or copy the DNA, make the messenger RNA. And then second, we're going to translate or read the code on the messenger RNA to make the protein. And we want to make sure that you know the location of where transcription and translation occurs in both prokaryotes and eukaryotes. And we're going to look at a picture of that in just a second. Okay, so just a picture, an overview of transcription and translation. So we start with our double-stranded DNA up here on the top. And we've got a particular gene that we're looking at here in the DNA. And then the first step, we're going to transcribe or copy the DNA, and we're just going to use one of the two strands of the DNA to transcribe, serve as our template, and then we'll make our messenger RNA. And then the second step, we'll translate the messenger RNA, we'll read the code, and figure out what amino acid to put in which order to make the protein. All right, so this is important. So this shows you the location of where transcription and translation is happening in both prokaryotic and eukaryotic cells. So this would be really easy for me to ask on the quiz and the exam, so make sure you know this. So prokaryotes are really easy. So they don't have a nucleus, so they're going to have both transcription and translation. Everything's going to happen in the cytoplasm because they don't have a nucleus. So both transcription and translation in the bacteria and archaea, everything's going to happen in the cytoplasm. So that's easy to remember. Eukaryotes, though, do have a nucleus where they store their DNA. And so transcription is going to happen in the nucleus because that's where the DNA is. And transcription, remember, is where you're going to copy the DNA and make the messenger RNA. And then translation is going to happen out in the cytoplasm because that's where the ribosomes are. So in eukaryotes, transcription happens in the nucleus, and then translation happens out in the cytoplasm. So they actually occur in different places in the cell in eukaryotes. Okay, we have got a couple of terms here. When we talk about the two strands of the DNA, we've got the template strand, which is the one that we're going to use as our template to transcribe and make the messenger RNA from. And the template strand we're going to read in the 3 to 5 prime direction. So we're always reading 3 to 5 prime. Okay, and then the other strand that we're not using, they call it the coding strand, which I think is kind of confusing. So we'll just ignore the coding strand because we're not going to be using it, and we're just going to be using this template strand, and that's the one we're going to transcribe to make the messenger RNA. Okay, now just a couple of terms here. Sometimes you'll see the template strand is called the antisense strand. So when you see the word antisense, it just means that it's complementary to the messenger RNA. Okay, and you can see that if you look. You've got your template strand, you've got a T there, and then on the messenger RNA you've got an A, so you've got A's and T's, G's and C's, and then obviously in the messenger RNA you don't have T's, but you'd have U's instead, so you have A's and U's here. So the template strand and the messenger RNA are actually complementary to each other. Okay. And then the coding strand, another term for that is the sense strand. So anytime you see that terminology, sense just means that it has the same sequence as the messenger RNA. So you can see this coding strand, you have an A, and then you have an A in the messenger RNA, and you have a C, C's, and you have the T in the DNA, but there's no T in messenger RNA, so you'd have a U instead. So anytime you see sense, it just means it has the same sequence as the messenger RNA. Okay, so if you take a genetics class, you'll already know those terms. Okay, so we've got a couple of terms here, and I have everything written down, but I think it'll just be easier if we take a look at a picture. Okay, so what we've got on our picture on the top, we've got the double-stranded DNA, and we've got the template strand here on the bottom in the light purple. Okay, so that's the one we care about because that's the one we're going to transcribe and use to make the messenger RNA. Okay, and the coding strand is the one we're not going to use. Okay. All right, and on the DNA, the start of the gene is going to have a special sequence in it, and we call that part of the DNA where the start of the gene is, it's called the promoter. Okay, and that's going to be recognized by the enzyme that does transcription, which is called RNA polymerase. So RNA polymerase will run along the DNA, and it recognizes that, oh, there's that special sequence. There's a bunch of T's and A's together in the DNA, and I know that that's the promoter and that's the start of the gene, and so I'm going to bind there, and then I'm going to start, transcribing that gene. Okay, and then it will transcribe the gene until it gets to the end of the gene, which is going to be indicated by another sequence in the DNA, and this is called the terminator. And so then the RNA polymerase will fall off and stop transcribing, and then that will be the end of the messenger RNA. So you've got the promoter just telling the RNA polymerase, hey, that's the start of the gene. And then you've got this terminator sequence in the DNA that tells the RNA polymerase that that's the end of the gene. You can stop transcribing. Okay. All right, and then we have this leader sequence in the DNA that does get transcribed into the messenger RNA, but it's actually not going to get translated. So this won't code for any of the protein. And there is a sequence in here called the Shine-Dalgarno sequence, which we're going to talk about later when we get to translation. That actually is going to be important in bacteria for translation. Okay, and we actually don't have this in eukaryotic cells though. And then we do have the start codon in the messenger RNA that always codes for methionine in eukaryotic cells, and in bacteria, they just have another little chemical group added on to the methionine. It's called N-formyl methionine. So the AUG codes for that. And then we've got the stop codon that will tell the ribosome, oh, that's the end of the protein. You can stop translating. Okay, so we'll get to that when we get to the translation. Okay, and then there's 3 stages of transcription and translation. So it has to start, so that's called initiation. And then we're just going to continue with the transcription or translation, so that's called elongation. And then termination is when the transcription or translation stops. And then this is just a nice little animation with a little tutorial there. That's pretty short if you want to watch that at home. And then I've got a bunch of other tutorials. Here's one on transcription. And then if you want to actually watch transcription occurring in real time and see actually how fast it happens in the cell, this is pretty cool. And then here's a tutorial on translation, if you want to watch that. And then this video here shows translation occurring in real time, so you can see how fast it happens in the cell, which is pretty cool.